Hello. I'm an AI plant manager at this CMO Centre, and I'll be accompanying your vaccine DS production VR program. Let's take a tour of DS production, shall we? Guest mode. Select your program. Hello. Welcome to the K-Bio CMO Centre. Let's proceed to the manufacturing facility entrance to start the GMP tour. This is the entrance to the manufacturing facility. Visitors must fill out the visitor list before entering the manufacturing facility. Here is the shoe cabinet in the locker room for storing outdoor footwear. This is the closet in the locker room, where you can keep your clothes and personal belongings. You can safely keep your personal belongings in the lockers provided in the gowning room. Please remove all clothing except for underwear, including personal accessories, tops, bottoms, and socks, and proceed to the central gowning room. This is the central gowning area for wearing clothing to enter the CNC zone. After washing your hands at the designated area, please put on disposable hair caps, disposable socks, and two-piece innerwear and wear shoe covers. Once you have finished putting on your clothing, please ensure that you are wearing the appropriate CNC regulation attire. This is the first gowning room, where you can gown in your attire before entering Clean Zone D. After crossing the area dividing line on the floor, put on disposable shoe covers and another disposable hair cap before proceeding to the second gowning room. This is the secondary gowning area to enter the Clean Zone C area. First, please put on disposable gloves and a disposable mask. Next, be careful not to touch the floor and put on shoe covers, clean room garments, and dust-free hood. Then, check your clothing in the mirror. Finally, sanitize your hands and enter the manufacturing area through the corridor. Now, we entered the culture manufacturing area. Let's proceed to the seed cell culture room. This is the cell culture culture room where the initial cultivation is conducted, which is the first step in the upstream process of vaccine manufacturing. Since cells have a growth stage, a large amount cannot be cultivated at once. Here, the cell strain is gradually cultured to adapt and grow in the environment according to the growth stage of the cells after growing the cell strain. In the cell culture room, human handling is possible up to a certain scale of operation. Now let's look at the initial cell culture equipment. A biosafety cabinet is a device used for aseptic cell culture work. It blocks external contaminated air to enable sterile operations and at the same time prevents contaminated air from flowing towards the operator. This structure protects operators from biological hazards. Inside the cabinet, manual work involving cell culture media, inoculation, and subculture is carried out aseptically. The centrifuge is an equipment that uses rapid centrifugal force to separate cells and culture media in order to obtain only cells. It is used to collect microbial bodies, separate and extract cellular organelles, and precipitate and separate culture media or solutions for the purpose of purifying solutions. With even stronger centrifugal force, it is also possible to separate difficult to separate substances in the body such as DNA, RNA, and proteins. The CO2 incubator is a device that maintains the appropriate temperature, humidity, and carbon dioxide supply for cell growth, allowing cells to grow well. It maintains a gas environment with added carbon dioxide to supply a certain level of carbon dioxide to the culture medium. This equipment is used to culture cells at a certain temperature and for a certain period of time. Workers can adjust and monitor the desired levels and conditions during the culturing process. The inverted microscope is a device used to observe the cultivation status of cells. Unlike a regular microscope, it has an objective lens mounted upside down, making it suitable for observing the shape and function of cultured cells growing on the underside. The inverted microscope is used to monitor the number of viable cells and cell viability. If the results meet the criteria, the next culture process proceeds. This is a cell culture room. As the scale of cell culture has become too large to handle manually in a general culture room, equipment-based cell culture is conducted in this cell culture room. Now let's take a look at the cell culture equipment here. The 20-liter wave bioreactor system is a device used to mass culture animal cells up to 20 liters using a disposable bag, meaning it is a scale-up equipment. It enables continuous and uniform shaking of cells in the culture medium by shaking the cell bag back and forth at a designated speed and angle, providing appropriate gas supply. 50-liter bioreactor system is a device for mass cultivating animal cells, up to 50 liters using a disposable bag. When the cells reach a saturation point in the 20-liter device, a certain concentration of cell suspension is transferred to the 50-liter bioreactor system to continue the culture under optimal conditions. For 50-liter cultivation, precise control of temperature, pH, aeration, and stirring speed can be achieved to allow cells to grow better. The 200 liters biological culture system is a device that uses disposable bags to mass produce animal cells up to 200 liters. When the cells reach saturation in the 50-liter bioreactor, the cell suspension of a certain concentration is transferred to the 200-liter bioreactor for cultivation under appropriate conditions. Because the cultivation period is longer in the 200 liters bioreactor, it is necessary to sample the culture medium during the period of cell growth and target substance secretion to ensure that nutrients such as glucose and glutamate are not depleted. If nutrients are insufficient, they need to be supplemented. Entered the purification manufacturing area. The area for handling viruses must be separated from the area for non-viruses, and the entrances and exits should be separated as well. When handling highly pathogenic viruses, additional disinfection procedures should be carried out accordingly. Let's move on to the virus culture and initial purification room. This is the virus handling area, the virus cultivation and initial purification room. The purification process of vaccines means the process of manufacturing safe vaccine drugs by recovering the maximum amount of products within the range prescribed for the manufacture of vaccine drugs, removing impurities. This process includes all processes from the recovery of culture media to raw materials for the production of finished vaccine drugs. Let's take a look at the equipment in this area. The process of achieving the final target cell line necessary for virus infection in a 1,000-liter bioreactor system is called cultivation. Once the cultivation is complete, a sufficient amount of virus is injected into the cells for a certain period of time to infect them, and the virus is then separated through a recovery process. At this point, a solubilization process may be carried out to remove cellular debris from the host cells to obtain pure virus or high-purity cultivation material. The first cell harvesting system used in the purification process is a filtration device for clarification during the rock recovery stage to separate the target protein and impurities. It is a depth filtration device that is equilibrated with a buffer. After the cell culture broth is fully dissolved, it is filtered according to the process conditions. Once the filtration is complete, the broth is separated and recovered. The ultrafiltration and diafiltration system is equipment that can be used to concentrate and desalinate proteins, as well as to separate vaccines and remove a lot of water. It is used tangential flow filtration and used to remove and displace cultures from cell cultures that contain buffer components, as well as to concentrate cultures to a desired volume. chromatography system is a purification equipment designed to remove impurities from vaccine compounds that have been concentrated and dialyzed by the ultrafiltration and diafiltration system. The column is first filled with a stationary phase resin, which allows it to be filled to a suitable height for protein separation and purification. It is the actual site where samples are separated. The chromatography system injects the protein-containing culture harvest or buffer, which is a buffer solution, into the column. The progress of the process can be monitored using various detection devices. The separation method using protein characteristics is commercially advantageous for vaccine production because it can process large volumes based on the size of the column and resin. This is the virus inactivation room. This is where virus vaccines are chemically or physically treated to irreversibly destroy and modify the virus structure, rendering them inactive. If viruses with infectious properties are to be commercialized as vaccines, they may cause disease in the human body. Therefore, virus inactivation is performed to ensure the safety of the vaccine. The virus inactivation system is equipment that artificially removes the replication function of the virus by applying heat or chemical treatment. Inactivated viruses are unable to replicate in the body and their infectivity is eliminated. This is the final purification room. Vaccines that have completed the virus inactivation process undergo final purification, just like the initial purification, using the ultrafiltration and diafiltration system and concentration system and the chromatography system. As the purification process progresses, the volume decreases as it is purified into the desired pure vaccine. As the product undergoes the next process, the size of the equipment also becomes smaller. Let's take a look at the final purification equipment. Like the initial purification stage, the virus inactivated vaccine intermediate is subjected to a purification process, using the chromatography system to remove impurities within product specifications and increase purity. Various resins can be used depending on the characteristics of the vaccine, and polishing resin, which is primarily used to remove trace impurities in the final stage, is used. The vaccine that has gone through the chromatography system undergoes concentration and dialysis filtration to achieve the concentration and formulation solution according to the raw material standard. From this process on, there is no direct change in the production volume, and it is a critical step that significantly affects the process yield and production quantity. Therefore, the process is carried out with caution to minimize loss. So far, we have learned about the upstream and downstream processes of vaccine production. Now, let's move to the exit area and go through the procedures for leaving the manufacturing area. This is the exit anteroom. Take off the dust-free clothes and place them in the recycling bin. First, take off your shoe covers, clean room garments, and dust-free hood, then put them in the recovery bin. Next, take off disposable shoe covers, disposable masks, outer layer of disposable hair cap, and disposable gloves, and put them in the waste bin. Then, go through the CNC corridor to move to the central gowning room. This is the central storage room. To enter the CNC area, please put your dust-free shoes and innerwear in their designated places. First, remove your dust-free shoes and place them in the shoe cabinet. Then, remove your innerwear, starting from bottom to the top, and place them in the innerwear cabinet. Remove your disposable hair cap and put it in the designated waste bin. Then proceed to the gowning room locker. This is the gowning room locker. Open the closet where you placed your clothes and put them on, and retrieve your personnel belongings. This is the shoe cabinet. Open the shoe cabinet, put your shoes on, and exit the manufacturing facility. This is the end of the GMP manufacturing facility tour. Please press the GMP tour and button to exit the content.